We are now going to be looking at the last section in chapter 7, which has to do with buffers. Buffers are going to be a solution that can resist changes in pH when acids or bases are added to a solution. It is interesting to know that biological systems, like the human body, actually have buffers in order to maintain the pH of the body. Specifically, in our blood system, the buffer system that we have wants to maintain the pH of the blood between 7.35 and 7.45 pH levels. What happens is the body changes pH, meaning that it goes into alkalosis, meaning higher than 7.45. You can see that changes like that can lead to death. But even if the pH goes lower than 7.35, we can see that changes in which we have lower pH in the blood can lead to also death. So it is important that the body develops specifically a system in which we can maintain the pH level at this range. Also, many of the biomolecules that work in our body need this pH because that's when they are most reactive and effective. It is important to note that when it comes to buffers, buffers are formed by combining equal concentrations of a weak acid and the salt of its conjugate base. To give you an example of a buffer system that is not in our body, but it is considered a buffer system, let's say that we look at HF. HF is a weak acid. The salt of this weak acid is going to be when you combine an element from 1A to the conjugate base of HF, meaning if HF loses a hydrogen, is going to make fluoride. So KF is going to be the salt of its conjugate base. Now, when we have a buffer system, these pair of compounds that are present in equal concentrations in a solution, one is going to react with the acid, with the incoming acid, while the other one is going to neutralize the incoming OH. Buffer systems are not always neutral. We can have buffer systems that regulate acidic pHs. We can have buffer systems that regulate neutral pHs. And we can have buffer systems that basically neutralize or maintain is the better word, basic pHs. Okay. Buffers have what we call the buffer capacity. And the buffer capacity is going to be a specific amount of acid or base that can be absorbed by the buffer without causing significant changes in pH. Okay? Three major buffer systems prevent the buffer capacity from exceeding specifically what happens in the human body. In the human body, we can have the bicarbonate buffer system, we can have the phosphate buffer system, and we can have the plasma that is present in proteins. It is important to note that typically the most common one that we talked about is going to be the blood buffer system, which out of these three is going to be the bicarbonate buffer system. Let's now look at it specifically. So in this bicarbonate buffer system, understand that carbonic acid is going to be our weak acid. And carbonic acid specifically has its conjugate base, which is HCO3 minus 1. The reason why the blood buffer system is called bicarbonate buffer system is because the conjugate base of carbonic acid is bicarbonate. What happens overall in this system is that because the system it is regulated by respiration, meaning the intake of CO2 is going to regulate overall the formation of carbonic acid and in turn the concentration of bicarbonate through respiration. Understand that this blood buffer pH could be regulated. Let's look at specifically figure 7.35 to look at this regulation. Okay. So I'm just going to zoom in slightly into this. Okay. So normally in the body, we're going to have a pH between 7.35 to 7.45. Let's say that all of a sudden, when we have a system that all of a sudden became quote unquote stress because there's changes in the environment. Let's say that the stress that the system is going to encounter is going to be decreased blood CO2. Okay. So, in a way, when we look at this buffer system, we can relate this concept back to Le Chatelier's principle. In Le Chatelier's principle, this is saying that in this case, if we decrease the amount of CO2, overall, the system is going to now push the equilibrium in the right direction in order to replenish this product that is lost. You see that the observation is that when you decrease CO2, there's an increase in pH blood. But in a way, that makes sense. Because since Le Chatelier's principle says that now the rate of the reaction in the forward direction is higher, we are going to combine more H plus and HCO3 minus 1, make carbonic acid to replenish that CO2. So that means that overall, when that happens, since we are not going to have plenty H plus around, that's going to result in an increase of pH. So now the pH is going to be more basic. Let's say that in a body, then the opposite happens. What about if all of a sudden we are going to be increasing the levels of CO2? If you increase the levels of CO2 according to Le Chatelier's principle, then what's going to happen is that now the reverse reaction is the one that it's going to shift the equilibrium. So the equilibrium, is going to shift in this direction. If the equilibrium shift in that direction, that means that we're going to have more of H plus. If you have more of H plus, then that means that more H plus means lower pH. Now, understand that when this happens, these changes can occur during respiration. So, a particular way in which you can actually decrease CO2 is by hyperventilation. In a way that you can increase CO2 level in the body is when people cannot breathe out. So, for example, they have some problems in the lungs that they cannot breathe out. That will be an instance in which CO2 levels in the body are going to be increased. So you can see that this interplay of buffers, we can also incorporate to it the Le Chatelier's principle because overall, this is an equilibrium reaction. The kidneys can also serve as a control for blood pH. The kidneys help keep our blood pH balanced by controlling how much acid and base we keep or get rid of. If we focus on figure 7.36, which illustrates the urinary control of blood pH in the kidney, we can observe that the CO2 from the blood enters specifically the distal tubule in the kidney. Then it reacts with water to form carbonic acid. In turn, this carbonic acid is going to break into hydrogen ions and bicarbonate ions. We see that the hydrogen ions are sent out into the urine to remove excess acid, while the bicarbonate actually returns to the blood to help neutralize it. You can see that the source of sodium, which is an element in group 1A, it comes into the kidney and it joins that bicarbonate anion to make a salt of the conjugate base for carbonic acid. This process helps the body to stay in healthy pH range, which is important for the things like, for example, enzymes or other biomolecules in the body in order for them to function correctly and for cells to have normal activity.